Thank you for the introduction, and thank you for inviting me to give a talk here today. Today I'm going to talk about our new vaccine platform for African swine fever, and I'm also going to give a little bit of an overview of what we've been doing in experimental ASF vaccines. As most of you know, about 40% of the world animal protein comes from swine, with top pork consumers being China and the European Union, and the U.S. at number 8, where China and the European Union eat approximately 50% more pork than we do. When you look at the heat maps of domestic swine farms, you can see that there's concentration of domestic swine is in areas of very high consumption. And when you look at yearly pig numbers, in the U.S. we produce about 133 million pigs, 271 million pigs are produced in the European Union, and over 700 million pigs in China. And when you look at it by graph form, you can see here in red about half of the pigs in the world are produced in China. And that brings us to African swine fever virus. It's the only member of the Asfv family. It's a very large double-stranded DNA virus. Has a linear double-stranded DNA genome of about 170 to 190 kb. There's over 150 genes in all genomes, with very few of these ever studied in detail. There's only about 20 that have ever been tested with any functionality tests. The initial cellular target is macrophages. There's so far no stable cell line for growth. When you adopt African swine fever 2 cell lines, you get very large genomic deletions. And there's at least 20 different genotypes. And here's just a nice EM picture that was taken at Plum Island showing the double membranes of African swine fever. And currently there is no commercially available vaccine. The disease is highly lethal to subclinical. It causes hemorrhage, edema, ascites, and shock. It appears in wild boar, warthogs, domestic pigs. The virus host factors responsible for different outcomes remain poorly understood, and the differences in strain virulence are not really known, including varying host species and symptoms and lethality. We know there's a very rapid and efficient transmission among pigs, and we know that there's a long-term persistent or latent infection that has an unknown frequency or duration. So how is African swine fever transmitted? We know it can be transmitted from domestic pig to domestic pig, from wild boar to wild boar, and from wild boar to domestic pig and back and forth, as well as with a soft tick carrier. And this soft tick is often found in burrows of wild boar. They're found ASF positive. Wild boar are very promiscuous and they share each other's burrows. It's a way of transmission. And it's also been shown that wild boar and ticks can be persistent carriers of an unknown duration. There's also a lot of indirect transmission with African swine fever. It can be several days in feces, months if the pens are contaminated. It's been shown to be around 18 months in blood and years if the carcasses are frozen. Pork products have shown positive ASF 140 days after they've been salted or dried, and also feed containing contaminating blood or feeding swill, garbage, and waste. And here's just a picture of some pork that was seized from an ASF positive country entering into Canada, and this was found earlier this year. And here's another picture from March where there was 1 million pounds of pork seized at the New Jersey port of entry that came from China. So African swine fever has changed a lot in the last few years. And when you look back to 2005, it was endemic in southern Africa and in Sardinia. And because it was mostly in Africa, it was not a concern for the world, and very few people were studying it. In 2007, there was a single introduction of African swine fever in Georgia. And over the course of about the next 10 years, this single introduction spread all the way into Poland and Eastern Europe, all the way north in Russia, and has continued to spread. But in the last year, African swine fever made its way into China. from China, quickly spread to Mongolia, Vietnam, where they've had hundreds of outbreaks. They had to cull 1.2 million pigs early on. It then entered earlier this year into Cambodia, Laos, Myanmar, Timor-Leste, South and North Korea. There's been multiple ASF-positive pigs washed up on Taiwan, and both Japan and Australia have had incoming ASF-positive pork products. ASF also made a jump into Belgium. Luckily, this was discovered early, and it was in a small, single area that was quickly quarantined. And there's only wild boar in this area, and now they're doing complete culling of wild boar to control the disease. So now we're in 2019, and the picture looks a lot different than 2005. ASF is definitely a worldwide problem. There's no vaccine, there's no treatment. It's rapidly spreading, and it's rapidly affecting the world's pig population. Some of the reports are about half of the pigs are dead in China, and their production is about half of what it was last year, which creates a worldwide protein shortage problem. Luckily, a few years ago, Cyril Gay actually came up with the idea to start a global African swine fever research alliance. And here's a picture from when we met in Italy in 2018, and our next meeting will be in Kampala, Uganda in 2020. And when we get together, we start talking about what are the gaps and what are the problems in African swine fever. And when we're talking about ASF, why is it spreading? There's no vaccine. It's only controlled by culling. There's a lack of reporting systematic animals, especially with small backyard farms. There's also the inability of small to medium farms to have any realistic biosecurity measures. Pig blood and byproducts were being used in feed. It's had uncontrollable wild boar pig movement. And there's also illegal or unmonitored domestic pig movement. So where are we with ASF vaccines, and what are the gaps? We don't know the protective immune mechanisms. Protective immunity from viral proteins are unknown. This makes it very hard to make a subunit vaccine. However, some people have tried, and there are some experiments that have been done with subunit vaccines. However, any attempts have failed or gave only a very modest protection rate. Some of the results are contradictory using different virus isolates. And protection that was achieved has been no more than 20 to 50% of the vaccinated animals. And all of these experimental subunit vaccines were at very high doses, which make them commercially unviable. So right now we've been looking at vaccines, and they've been mostly live attenuated vaccines, which rely on the development of recombinant African swine fever. And this has been a very time-consuming process. And we look at the timeline for development recombinant ASF vaccines that was actually started in the 90s by Dan Rock's group. We found that for us it was taking about 2 to 3 months to make our recombinant plasmid, another 1 to 3 months to get recombination to work, another 3 to 10 months to start getting plaque purification, which could take up to 30 passages. A couple more months to produce a stock and check to make sure the absence of parental virus by next generation sequencing and PCR, and a few more months to check for virus attenuation in animals. And when this all came together, it was taking about 2 years from concept to completion. So the question asked was, could we find a faster alternative for creating recombinant African swine fever in field strains? And in the past 20 years or so, there's been a lot of new technologies, and we decided to implement a couple of these. We introduced fluorescent markers to allow for faster purification, and we started using CRISPR-Cas9, which resulted in a 4-log increase of recombination. And just doing those two simple things, we were able to cut down almost every step of the way to make recombinant virus and get to an area where it takes us about 3 to 4 months from concept to recombinant virus purification. And that brings us to making our live attenuated vaccines. And our initial attempts at live attenuated vaccines was taking some of the information from the 90s, again from Dan Rock's initial studies in ASF, and taking some of these attenuation factors and putting them into the new outbreak strain, African swine fever, Georgia. The first one that we did was 9GL, and when we deleted 9GL from African swine fever, you can see here that at 21 days at 10 to the 2, we had partial protection, but we had full protection at 10 to the 2 for Delta 9GL. So we could see that this was protective at a single low dose. However, when we increased our doses of our Delta 9GL, we saw that the virus was our vaccine was actually virulent. So sublethal doses of ASF Delta 9GL induced protection against clinical disease and death caused by ASF-Georgia. However, deletion of 9GL did not produce the attenuation effect comparable to that induced in Malawi or Pretoria strains. So with this, we knew that we needed to come up with a way to get further attenuation of our live attenuated vaccine. And here, what we did is we introduced another attenuation factor, this gene called UK. So we had two separate deletions in this live attenuated vaccine. Introducing UK into our 9GL platform, we were still able to grow the virus in macrophages, although it was slightly reduced. When we challenged with this new vaccine strain, we found that we were able, again, to get partial protection at 10 to the 2 and full protection at 10 to the 4 and 10 to the 6. But importantly, deletion of UK in the Delta 9GL backbone increased viral attenuation at least 100-fold and induced protection when administered once at 10 to the 4. This onset of protection started at 14 days, which was the fastest that we've ever seen protection with any African swine fever live attenuated vaccine. And this is the only double deletion that protects against current outbreak strain African swine fever, Georgia. The next thing we looked at was deletion of a group of genes that we call Delta MGF, which is actually deletion of 6 genes in the MGF family. And this deletion virus was, again, protective at a low dose, about 10 to the 2, and we didn't see any clinical symptoms at higher doses, and we only saw 1 day of mild fever. And this Delta MGF platform is obviously pretty popular because several other groups have used the exact same deletion. This has been in by Pirbright, and recently 2 groups in China, Harbin and the Institute of Military Vaccine Medicine, have also created a Delta MGF backbone in ASF-Georgia. It's important that all these MGF platforms contain the same deletions as in the USDA 2015 platform. However, the MGF region has been widely shown to be unstable, and ASF can adopt to favor virus replication under selective pressure. And this gives the potential for any ASF Delta MGF strain to recombine with a circulating strain of African swine fever and result in a vaccine that becomes virulent. So we know that MGF is a good attenuation factor, but you need an additional safety factor. So what we did is we combined our MGF and our Delta 9GL, and when we did this, what happened was the virus was still non-virulent, but it was also non-protective. And this shows that additional gene deletions can further decrease the ability of virus to replicate in swine, which can result in inefficient antigen presentation and decreases the ability to induce protection against challenge. The 2 groups in China, what they did is they deleted a gene called CD2. CD2 was first identified in 1992. It mediates heme absorption. There's been natural isolates that have been found with deleted CD2. However, most of the natural isolates have other genomic mutations present and are only attenuated at very low doses. And some of these are virulent at higher doses, and they do create a non-heme absorption virus. So we wanted to look at and see, is CD2 in Georgia, if we just delete CD2, does it have any effect? And the answer is it doesn't. Deletion of CD2 in ASF-Georgia has a disease that is indistinguishable from wild-type virus. And you can see here both whether we gave low to high dose of intramuscular or intranasal, the outcome of the disease remained the same. So when we're looking at the delta MGF platforms created around the world that rely solely on the delta MGF for attenuation, we want to remind people that delta MGF area has a high rate of recombination and it has caused virulence before. Additional virulence factors that attenuate ASF should be included for safety, and for those groups, we just want to say be careful in pursuing any ASF vaccine based solely on this one deletion for attenuation. So I've talked a little bit today mostly about some of the production of historical determinants of virulence and deleting them in the current outbreak strain. And these deletions were successful in attenuating African swine fever Georgia, but we still have a need for safer and more attenuated vaccines, which led us to how do we identify several new African swine fever protein targets. And we used a custom computational pipeline that takes into account different sources of bioinformatics and functional genomic data. And by doing this, we were able to categorize and prioritize which genes could be potential determinants of virulence. And we started with a gene called EP152R, which was predicted to be a CCL2-like protein, and this was essential and cannot be deleted. We also looked at L83L, which was an interleukin-1 binding protein, and this deletion did not fully attenuate the virus. But today I'm going to talk about a new gene, and this gene is I177L. I177L is located on the reverse strand of the virus. It's highly conserved among all isolates. When we look here, you can see on the bottom, I177L is a late transcribed gene. And it's a gene that, through our pipeline, was predicted to be an immune modulatory protein. We found that I177L is not an essential gene. We were able to delete the gene and replace it with P72M cherry cassette. And we found deletion of I177L affects virus growth in macrophages a little bit. However, we can still achieve high titer stocks. When we looked at I77L and we put different doses in, we found that at all the way up to 10 to the 6, there was completely attenuated in swine. There was no clinical disease. There was no fever. And the highest degree of attenuation was achieved for any recombinant African swine fever Georgia. When we challenged our animals with animals that are vaccinated with deletion of I177L at 28 days post-vaccination, we found that 10 to the 2, or a single dose, could protect against challenge against African swine fever Georgia. That African swine fever Georgia Delta 177L is the most potent single low-dose ASF vaccine platform. We saw no clinical disease after challenge, including no fever. And when we looked at viremia, we saw that we could see apparent sterile immunity, that we had no measurable challenge virus replication at 10 to the 4. When we looked at different doses and we looked at viremia, we found that even at low as 10 to the 2, we were able to see viremia of our vaccine and antibody response. And it's important to note that the antibody response was achieved at about the same level regardless of dose. We then looked at viral shedding, and when we did this by adding a sentinel pig to be housed and co-housed with all of our vaccinated animals. And you can see here in the red line is our sentinel pig, and we saw no detection of vaccine virus. So this means there's no functional viral shedding of our vaccine Delta I177L. So this vaccine virus does not spread to unvaccinated animals, and this is important as this increases the safety profile for this vaccine. So in conclusion, with our new vaccine platform, we saw that we had the highest degree of attenuation for any live attenuated vaccine that protects against current outbreak strain African swine fever Georgia. We had complete protection even at a dose of 10 to the 2. So that lacks residual virulence even at doses as high as 10 to the 6. This vaccine does not induce viral shedding, induces apparent sterile immunity, and has the highest safety profile of any live attenuated vaccine that protects against African swine fever, Georgia. So what are we doing now? Well, we have 3 functional single-dose live attenuated vaccine platforms. We have licensed our Delta 9GL, Delta UK, and our Delta MGF platforms. For our I-177L, it's still new, so we're seeking a commercial partner if anyone has a few $1 million to spare. We have a commercial partner to develop a DIVA platform and our double deletion platform, which will include safety testing, reversion to virulence, development and certification of a DIVA test. And right now, an introduction of DIVA in all of our platforms is underway. We have 6 DIVA targets that have been identified. We know at least 2 are non-essential, and we've confirmed a strong immune response in serum samples from ASF-positive animals. And our preliminary results confirm that these DIVA targets can work by ELISA. However, we know that a DIVA-compatible live attenuated vaccine is probably still years away. So just to conclude and to recognize all of the people in African swine fever that helped, obviously Manuel Borca, who we've had a good partnership and have been running the African swine fever program at Plum Island. I'd also like to point out a couple people. Sarah Pruitt's been making all of our macrophages for these studies, and Elizabeth Ramirez has been a recombination machine and produces quite a few of our recombinant viruses, and we're able to achieve at least 1 per month, sometimes even up to 3 or 4 per month. And finally, we're doing a special issue in viruses on endemic and emerging swine diseases, so hopefully a lot of people in this group, we welcome your submissions. And finally, if you're a virologist, it'd be great if you joined the World Society for Virology. It's free, it doesn't cost you anything, and it's an effort to try to get virologists around the world together. And with that, I'll take any questions.
My first question is, do you have any information on heterologous protection against other genotypes?
Right now we don't. We're mostly concerned with the African swine fever, Georgia, which is the same strain affecting all of Asia and Europe. So that is something that we want to do in the future.
Doug. Hello.
Yeah, we got you.
Okay, I think the first question that I had already was formulated by Bob. Does it make sense to look for a multivalent protection? I mean, it's important at this time, or do you think that just acting upon serotype 2, I guess, right, or genotype 2, right, is sufficient? And the other thing is, it looks like you have a dream vaccine, you know, from the standpoint of a sterile immunity and very, very good attenuation. What happens with that horror stories that existed from using modified vaccines in long terms and chronic immunological, pathological? Is that a possibility still with this safer vaccine?
Those are both really good questions, and when we're looking at cross-protection right now to control in Asia and Europe, we're probably okay with a type 2 vaccine. And with cross-protection right now, nobody has any idea of how many different genotypes or different strains are circulating in Africa, so obviously those studies have to be done. And if we're looking for worldwide eradication or to try to have something in case another introduction from Africa occurs, we obviously need to do those cross-protection studies. But we need to find out what viruses are actually out there and being circulating right now. For your second question, of course, long-term effects of live attenuated vaccines is a concern, and it is something that has to be looked into. But for right now, we're looking at just short-term studies and as a way to control a potential outbreak that could occur in the States or that's occurring in Asia right now, and a short-term solution rather than a long-term vaccination strategy.
Thank you.
Any indications of ability to replicate on immortalized macrophage cell lines with these different viruses that you've created?
We've tested a lot of the immortalized macrophage cell lines that are commercially available or that are available through labs that have made them, and so far none of them have been shown to replicate African swine fever, at least at a reasonable scale.
So Doug, great, very nice talk, congratulations. So if this vaccine is licensed by USDA, my question is, how will it be produced? We know all these viruses we produce, these recombinant viruses, we need primary cell lines, alveolar macrophages and similar cell lines to produce these vaccines. So even if we have a very safe, efficacious vaccine candidate, how can we produce enough doses, even if you go at 10 to the 3 or whatever, you still need to produce it in primary cell lines. How will we manage that?
Most of our commercial partners are not worried about that. Commercial entities have a lot of proprietary cell lines and a lot of production cell lines, and we're hoping that one of those works, but obviously that's something that comes in their hands at that stage.
So did you test then the stability, the genomic stability of your vaccine, producing it in some of these proprietary cell lines?
Commercial entities don't want to give us their proprietary cell lines, so it's going to have to be up to them to produce a master seed that we will then test and look for genomic stability at that time.
So I think some great work here and good news for those of us out in the field. But the question would be, how long would it take to get this vaccine to the field, if it could be licensed?
I think that all depends upon the regulatory authorities and what happens. For the U.S., it's a very different situation if we have an outbreak versus right now and what would be tolerable. We are looking into making a large batch in primary cells as an emergency response vaccine. So a lot of that depends mostly on regulatory authorities and to the country that it's going to. So I can't give a great answer to that question.
Okay, we'll move on to next talk. Thank you.